For the next topic in comparative genomics, we're going to be talking about metabolomics. Um, After we finish talking about this topic, hopefully you'll be better able to recognize metabolic networks and the corresponding graphs that are associated with these. So that include nodes and edges that reflect the metabolites and reactions associated with them. You'll also be able to understand the structure and dynamics and evolution of metabolic networks, recognize some of the ways in which metabolic pathways differ among different species, know how to organize and reconstruct metabolic pathways of an organism from its genome, transcriptome, proteome, and understand the general alignment, how we can generalize the concept of alignment to metabolic pathways. Understand the physiochemical basis of enzyme catalysis and the quantities needed to characterize enzyme kinetics. So in other words, how do we actually model this system? And appreciate some of the difficulties in modeling the traffic patterns of metabolic networks. Metabolomics comes from the study of metabolism. And when we talk about metabolism, what we're talking about are the flow of molecules and the energy and the flow of molecules and energy through pathways of chemical reactions. Specifically things like how to convert food and fuel to energy for cellular processes or cellular building blocks, or how to get rid of waste. When we think about metabolic reactions, what we're considering are the metabolites that are contributing to these reactions. Specifically, we're going to talk about metabolites as molecules that undergo a transformation in a biological system. And this typically happens either via enzyme catalysis or spontaneous reaction. This could include both small metabolites, such as sugars and amino acids, and larger metabolites, such as proteins and nucleic acids. And one of the key components of a metabolomic investigation is the identification of and the understanding of metabolism and how metabolism works, is understanding a metabolic pathway, right? So that's kind of what's going to underlie this process, because typically these metabolic pathways involve a series of different steps that may connect in a variety of different ways. And what this metabolic pathway is it's basically a roadmap that defines the possible transformations of various types of metabolites and the various states that they might reach. And conceptually, this pathway will form a network, and this network is represented as a graph with the metabolites as the nodes and the reactions as the edges. I think it's unfortunate. We often, as biologists, I don't think have a great appreciation for the role that graphs play in our understanding and modeling of systems. But increasingly, the applications of graph theory and graphs as models for how systems work is underlying more and more the ways that we analyze data. And you'll see in both the metabolomics discussion that we're going to have today, as well as when I talk further about systems biology, it's really important that we begin to shift our ideas towards these graph theoretical approaches to how we conceptualize biological systems. It really is becoming one of the dominant tropes in ways that we understand them. So when I'm referring to metabolomics in the same way that I'm referring to genomics and transcriptomics, right here we're looking at the comprehensive and synoptic view of metabolism. And what we're specifically trying to figure out is how we can integrate the integration of all the enzymatic activities and flow of metabolites that we can see in a single example. This is a very complicated and difficult question, but metabolomics, really, this area of study is incredibly important when it comes to thinking about how biotechnology and the industry of biotechnology functions. This is really the engine that is most important for our understanding of things like genetic engineering, for the synthesis of products, for pharmacology, incredibly important field for us. I think it's important that when we think about metabolomics, that we frame it as, how do we actually conceptualize and compare problems in metabolomics? And this is kind of a difficult way, because when we start to think about, how do we actually compare different graphs or different networks, intuitively it may seem really complex, right? But in this case, I think, we can see a very straightforward example, right? So in this case, what we're looking at is an example of a sequence alignment. So say we have a sequence alignment here where the sequence is A, B, C, D in one case, and in another case it is A, B, Q, C, D, right? And in this case, what we can think of is each sequence is actually a linear network, right? Each letter in this sequence, in either the first sequence on top or the one on the bottom, represents a node and an edge that would connect each one of these, right? So that they're connected in a line. And so when we align them, we can start to do some easy comparisons between what is different between these two sequences. And in this case, we have an insertion of a Q. Whereas if we look at this set below us, right, we can see that the network has changed. And how has the network changed? In this case, the C and the B has been inverted, right? And so now the network that we're looking at has changed. And so if we think about the connections and this alignment as a linear process, right? We were to compare these two, we can start to see a way, using these arrows, that we can compare the differences in how these alignments work based upon a network conception of them. And typically when we think about sequence alignment, what we're actually looking at is something where we have some sort of relative order constraint. And this is not unlike how we think about metabolomic networks, where the order of things actually matters, right? You can't have a final product metabolite without all of the precursors. So in that sense, there is sort of a flow or a linear process or a directional process that's involved, right? And some metabolic processes are incredibly linear. Others of them have much higher levels of dimensionality, meaning that the pathway is not simply, there's not just one way to get from point A to point D, right? There are multiple ways that we could go. And when we think about how we model these systems, what's different between sequence alignment and these networks is we're no longer modeling the pathway as a line. We're modeling it. The model that we're using is one of a pathway or a network. And so what we're thinking about is situations in which the model that we're using is non-linear. So it's not just a single line. And anytime we do anything but non-linear in the sciences, it's going to be more complicated, just how it works, right? So when we think about pathways, like what are our options, there are a couple different things we could consider, right? So pathways could be linear, they could be closed loops, they could be highly branched and densely interacting, right? But they don't. They could be all these kinds of things. They could also contain recognizable clusters or blocks. So they could be modular in their nature. We could look at these clusters or blocks and we could look for things that are both high density. In other words, they have a lot of connectivity among the nodes, a lot of edges coming out of these nodes, or they could be low density, where they wouldn't have very many different edges coming out of these nodes. And the more sort of connections or edges that you have coming out of the nodes, the more dense and possibly more complex your network could be, as there'd be more in various pathways that you might be able to go down. Other things about pathways, so in terms of all this other diversity, is they can also span species. So we don't even have to think about pathways being related just to just one species. So in this case over here on the right, like we can see some co-parasites of the glassy wing sharpshooter, right? And what we see in this case is that we've got Solsticia muelleri and Baumania psychedelinicola. And what we see is that there are, if we look at, say, like the amino acids that are synthesized by these various coparasites, we can see that, not the complete set of amino acids are created by either one of these species. So in order to get their full complement of the things that they actually need each other. They need to be able to create things and exchange these things. And so these networks, these metabolic networks, are actually intertwined across species. So not only do you have the metabolism of this species that's needed to create all these various items, but we actually need specific building blocks in order to create the things that the other one might need, and vice versa. So we can start to see how networks can be incredibly complex and link across species and link across organisms and even in cases such as these. So just a good example of how complex metabolomic networks can be. So let's start off by thinking about, how do we investigate and understand metabolomic networks? Well, one of the dominant ways that we do this is by looking at, the principal way we do this is by looking at protein function. And there are generally sort of two different perspectives we could take on protein function. There's one which looks to basically explain the biochemistry of the cell via the explanation and descriptions of the different functions of different enzymes. And another case which would more be related to the regulation of enzymes and other protein players and other functional molecular players by understanding the regulatory networks that control these biochemical reactions. So if we're going to try to get to a common understanding of how these protein functions and the biochemistry of these cells work, we have to come to a common understanding of how we talk about them and refer to them. And remember, this information is incredibly dense. There's a lot of detail and there's a lot of complexity in here. So it becomes really important that when we talk about things that we're all using the same language. So in this case, there's actually been quite a bit of effort to generate databases that are used for annotation, for establishing this additional information about function and purpose and biological activities associated with various types of chemical reactions or other sorts of activities. And so some examples of these databases include Enzyme Codes, Gene Ontology, KEG Maps, Interpro, and the Protein Database. And each one of these sort of takes a different perspective on what is sort of the way that it's going to address this question of protein function. And so we'll start by looking at the Enzyme Commission or the Enzyme Codes. And these are commonly applied, and I think it's a really good example that can show us sort of how we might take something that seems as difficult to understand as sort of this random 4 numeric field identifier and realize that it's actually related to something else, right? But so if we look at something like the enzyme codes, we can see that a common enzyme code would be something like this, where it's EC 1.1.1.1. And you might say, well, how am I supposed to understand this? Well, this enzyme code corresponds to the following reaction, which is an alcohol plus an AD plus. And AD plus is going to yield a corresponding aldehyde or ketone, and then an NADH and a hydrogen atom. So that's generally what this enzyme code refers to as versus this type of reaction. But it's interesting that there's going to be several reactions, different alcohols. Remember, it says an alcohol, not a specific alcohol, that would share this type of reaction. And so when we look at enzyme codes, we actually see that it's this first number which refers to a general class of biochemical reaction, right? Whether or not we're looking at enzymes that are associated with oxoreductases, transferases, hydrolases, lyases, isoamerases, or ligases, right? Then these second and third fields are dependent upon subcategories. So in this case, for oxiductoreductases, right, the second number describes what substrate you're targeting, and the third number would describe the actual acceptor of that reaction. And finally, field 4 would give some information about the specific enzyme activity. So the example here would be that if we shifted the component of the reaction, we would, instead of having it be EC111, we would have it be EC1112, right? Because we have a very specific change in how the enzyme activity functioned. How those enzyme activities can applied can be sort of viewed here in this KEG map. And so KEG stands for the Kyoto Encyclopedia of Genes and Genomes. And this map is a map of the valine, leucine, and isoleucine biosynthesis. And you can see that along this process, right, we can start at various places. We're going to start with from glycine, serine, threonine metabolism, right, and it'll walk us through the various sub or intermediate metabolites until we get to isoleucine biosynthesis, valine biosynthesis, or leucine biosynthesis, which then leads us to a protein, right? So it sort of shows this process, but notice that there are different ways that these items can connect to each other, and different pathways that we might go down depending upon where we start and what are the items that we have. And there are also possibly some different feedback loops that can have an effect on whether or not we go down one way or the other. One thing to note is that this is an example of an oxo. We can take just a very specific example out of this whole process, right? We could focus in on this oxidative oxoreductase reaction, right? So again, it's starting with a 1 in this position, right? Where we have an alcohol, right, plus NAD plus, which is going to yield an aldehyde or ketone, right, plus NADH plus a hydrogen atom, right? Very similar to what we saw before, but with some added specificity in this case. Right, if we were to look just at this one reaction, right, this one type of enzyme as described here by this number, right, there are 2 known orthologs, so that we can find that can be associated with that can accomplish this reaction or involved in this specific step, which then translates to about 6,240 individually identified KEG genes, and these are genes that are related to those orthologs that were found, that are annotated as such and found in NCBI or RefSeq or some other sort of well-curated database. Additionally, there's about 76,000 KEG M genes, or genes that have been identified from metagenome or environmental samples that could be identified as being having this specific function. And there's also even about another 15,000 KEG ref genes that have been identified in other microbial organisms that could also be contributing to this type of process, but haven't been annotated as such, right? So there is, it just goes to show you that how many different ways these types of categories could be accomplished, in that it's really important to understand that just lots of different things could have the same enzyme codes, but it's that they're related to this biochemical reaction, not necessarily this specific position. Like, there's not just one thing that fills this role, right? So this describes a biochemical reaction, not the actual enzyme sequence itself. So I mentioned, and I showed you just an example right now of a KEG map, or which in KEG again refers to the Kyoto Encyclopedia of Genes and Genomes. One thing about KEG is that it's incredibly dense, right? So when you click on something, each one of these links has lots and lots of things attached to it. I just said there could be 76,000 metabolic genes, metagenomic genes associated with any one of these spots, right? But they can be really, even though they're very dense, if we think about this, like, these are some great ways for us to compare or to ask some different types of questions. So, for instance, if we're going to think about things, do simple pathways evolve to be more complex? So how might we go about doing that? Well, we could evaluate and look and say compare genomes and see whether or not we have, more levels of duplication in some organisms versus other organisms, which might indicate that there's a higher level of complexity. Additionally, we could look and say, when we look and compare two different genomes, do we see representations of all of these different reactions? And if we don't, if they're missing someplace in between, right, then we would need to know, then we could use that as a way to try to identify, well, they must have evolved or have some sort of novel solution to deal with the fact that they're missing something in this place, right? So there's some really interesting ways we can use these networks to sort of frame our comparative questions. Another one of the dominant ways that we define function, that we are used to sort of describe functionality in these systems, is the gene ontology. An ontology is basically an annotation system, right? A system to make notes about something, right? And in this annotation system, one of the key parts of an ontology is that we've created a controlled vocabulary that can be applied, in this case, to all organisms, in all cases, right? So otherwise, think of it sort of like a species taxonomy, right? It's important that we're all using the same names of families and orders and classes and things of that sort, so that people, when they talk about what one species is versus another, we can all have the same understanding of what its evolutionary history is. If we didn't have that, it'd be very difficult for us to decide, like, one thing is one taxonomic group versus another taxonomic group. And so in this case, we apply this sort of ontological perspective to describe gene products, specifically protein and RNA, in any organism, and be able to understand certain components about it, right? And so there are sort of like three major branches in the gene ontology. These include molecular function, biological processes, and cellular components. And a lot of this work actually came from folks in the Drosophila melanogaster community, and they looked at KEG and EC and they thought, oh, that works great, but it only works for enzymes, right? It's basically only describing these biochemical processes, and they wanted to have a broader understanding of how to understand the function of genetic elements, right? So that's where the GEO comes from. And when you think about how molecular function works, this is basically describing things from the biochemist's point of view, right? From what is the elemental activity or task of a specific protein, Whereas when we think about biological processes, one of the added advantages of this is it's more from the cell's point of view, right? What are the biological goals or objectives of a specific genetic element? And finally, there's also a component which talks about the cellular components, because it actually matters the location or complex that's associated with a specific protein or genetic element. The way that gene ontologies work is that they're generally functioned as direct acyclic. They are structured as direct acyclic graphs. And over here on the left, you can see an example of a molecular function direct acyclic graph and a DNA and a biological process direct acyclic graph from the gene ontology. And what you can see here is that there are different hierarchies or levels within the gene ontology in both the molecular function and biological processes, right? And each of these sort of levels indicate sort of categories and subcategories. We tend to think of them, each node, as being the function or term, right? Whereas these nodes indicate some sort of ancestor-descendant relationship. So in other words, if we have a helicase, right, we could either have a DNA helicase or an ATP-dependent helicase, right? And so that and if we had DNA binding, we could have examples where we had chromatin binding and a DNA helicase, and they would both be doing protein binding. So, when we think about DNA-dependent DNA replication, right, there's a whole bunch of different subcategories of that. Right. And so in this way, we have sort of like these nested categories, which are not always mutually exclusive, right? Like we can see that sometimes like an AT-dependent helicase could be associated with two different types of enzymes, either a helicase or an adenotriphosphatase, right? But they are hierarchically related. There can be sort of this ancestor-descendant relationship between these different nodes. If we were to compare sort of how this might look from a enzyme code to geo comparison, we can see that they're not all that. There are definitely some similarities here, right? We have some general categories. We have a more general category here of catalytic activity, because basically almost all enzymes are going to be involved in have some sort of catalytic activity, right? And we can identify a specific type of that catalytic activity, in this case isomerase activity or isomerases. We can see, we can start to get down to these subcategories. Here we've got intramolecular isomerases, intramolecular oxidoreductase activity, and then we can see, as we get even more detailed, now we can have enzymes that transpose the CC bond. We can have an intramolecular oxidoreductase activity, right? And so we have their own codes at each of these different spots. And then finally, we get down to the last sort of specific task, right? And so they can be very similar. And one of the nice things is that this works well in both these cases. But if we're dealing with something that's not necessarily an enzyme, that's where things can get kind of complicated. So one thing to understand about metabolomics is that there are a lot of databases out there with different metabolic pathways. There's one that's just for E. coli, right? And this contains all the genome, transcription regulation, metabolism, and membrane transport just for E. coli. There's also BioSic, which is kind of like an umbrella collection of as many of these things as possible. Again, I've showed you the KEG Kyoto Encyclopedia of Genes and Genomes. And I think one of the things that is a common feature, though, when we look at these metabolic pathways, again, we talked about this before, is they're typically not completely linear pathways, right? Like they're much more complex than that. And so one thing to understand is that when we're looking at metabolomic questions and metabolic questions, right, they are going to be rife with various types of feedback loops. Either negative feedback loops, where after we create a product, it can feed back into the system to repress further creation of that product, so until it's used, right? or it can act in ways to promote steps further down the line, right? So as we create something, it can either repress one product, or it can allow for the expression of something else. So if we look over here, for instance, on the left, right, we can see that we have methionine synthesis from L-aspartate, right? And this is a 7-step process. When we get to step 1, just so you know, there's like 3 different aspartate kinases in this case that are all regulated differently too. So this is even just a generalization of those. So even going into this network is more complex than is represented here. But say we get down to step 3, we find that step 3 as well, 1, 2, 3, this step right here, this is the only way to get into this position of this process as well. Is step 3. There are both methionine and threonine biosynthesis pathways that can hop into this process. So it's a very complicated system where, even when we simplify it to something like this with these feedback loops, there can be a whole lot of other things that lead into this process. Even if we look at CRP, we can see here that there's almost 200 genes that help regulate CRP. But I think the important thing here is to understand that, there's a lot more complexity than we're representing here, but that there are these feedback loops that act to sort of complicate this process, to delinearize it, to make it more complicated. And this is an example too of the various ways that aspartate and threonine can regulate this process. So if we were to make methionine, right, this process has a negative feedback loop here. If we were to make L-threonine, we're going to have a negative feedback loop up here. And if we're going to make lysine, that can also limit this reaction, right? This can also serve as a negative feedback, right? So there's all different ways these reactions become very difficult to say that they're not just a linear process. Other things that we can notice about these pathways is they don't always work in a way in which we get to the same product in the same way, right? And so it's important that when we think about metabolism, like we're often interested in trying to figure out, like, well, why are some things different and why are some things similar among species, right? And they could have a similar evolutionary history, a shared history, and then that's why we observe the things that we observe. But other times, what we're going to do when we try to account for these differences is investigate whether or not, we see differences in the sequences, differences in the presence and absence of genes, difference in the cofactors that are available to that organism, and differences in these actual pathways. So are the pathways truncated? Are they extended? Are they the same type of pathways, right? And one of the big questions that we're often looking at is looking to see whether or not these divergences in metabolic networks are corresponding with species divergences. So are they associated and are they actually explaining the evolutionary history of a set of organisms, or is it that they've done something unique or something else in response to metabolism? An interesting example here is if we look on the right and we examine the 2 glycolytic pathways, the Emden-Meyerhof and the Ender-Dodorov pathways. We're going from glucose to pyruvate in both cases, but in the 2 we see that these are slightly different. They're going to be divergent pathways by how we get from one point to the other. These are not the same pathways. They have different players and different processes by how they get from point A to point B. but they're accomplishing the same task. And this is actually a great example of convergent evolution. We can even look more closely at the enter-Todorov pathway, and we can even see differences within different archaeas. And so if we look at halophilic archaea and thermophilic archaea, right, we see that it's not, we can see two very different pathways, sort of divergent pathways by which you would get from glucose to pyruvate. We don't necessarily see the homologs actually doing the same activity in these cases. They can change dramatically, and even the enzyme kinetics can change sometimes, while topologies can remain the same. So there's a lot of variation that can happen in these metabolic processes. So going back to these questions that we had identified earlier, how do we account for the differences and similarities in metabolism among species, and do divergences in metabolic networks correspond with species divergences? Especially when we're thinking about the second case and from a comparative genomics framework, we can end up with these alternative hypotheses about whether or not, the divergences we're seeing are due to static binary case differences, say like comparing the two different pathways that are going on between halophilic and thermophilic archaea, or maybe it's different kinetic constants that in these reactions that are driving things between species, or different regulatory factors. And one of the things that we're always kind of looking at in this case are the dynamics of traffic through these networks, or how much are they varying, because they can vary pretty significantly. And is that what's actually going to drive us with our ability to identify which of these three alternatives may be contributing to this factor? And if we think again about how would we investigate this? We could think about something like the simplest case, which is the something like the TRP operon, which is involved in the amino acid in E. coli, which is involved in amino acid tryptophan synthesis from chorusmate. And in this case, the operon gene order drives the process. So this is the most simple case of of our linear network, right? Where we have our promoter region or control region. We have our first reaction, which involves TRPE and TRPD. Our second reaction, which involves TRPD. Our third reaction, which involves TRPC. And our fourth and fifth reactions, which involve TRPB and TRPA. Right, and this is a classic case where we move in the operon sequences in a specific order. We can look at other organisms, and as long as we can use this sort of linear process, if we know it works linear in one case, to compare it to another case and see is that also linear? What are the changes in these reactions that we see between them? What are the changes in the regulatory factors that are controlling this? This is that simplest case. This is that line, that sequence comparison that we talked about at the beginning of this presentation. So, however, sometimes things are missing, right? That we may not see a network which is functioning the same way. So if we look at something like Methanococcus janischii, right, and it is lacking a shikimate kinase in this chorusmate process, right, processing process. And so where did it come from? So if we look at the genes involved, there's no sequence similarity with these, right? But what it looks like is that there was some sort of non-homologous protein that was recruited to fulfill that same role in this pipeline, and so it no longer, it's another convergent mechanism, right, where even though it's not expressed in the same way, the network that we're looking about has now diverged away from sort of this linear conception. And that's one way that we can start to think about, how we're going to drive and look at these different types of comparisons. So, suddenly we see the complexity changing dramatically, where we have something inserted or something coming up. And again, just to remind you, when we think about how we're going to compare these networks, we think about these questions about relative order constraint. We think about whether or not the process is linear or of higher dimensionality. And we think about if it is nonlinear, are we thinking of it as a pathway or a network as opposed to just a line? One thing to remember when we think about metabolic pathways is that metabolic pathways are dynamic, they're compensatory, and they're also responsive to their environments. So one of the ways that we might think to investigate them is to think about, well, what happens when we remove a specific component, right? So can we predict what's going to happen to that reaction and the metabolites involved in that reaction over time? And this is a way that we can start to understand, what are the key components contributing to various metabolic pathways and how important they are in the outcomes and what are the relationship between these entities. There are some different ways to do this. One of the ways to do this type of investigation is to think about doing a steady state experiment, where what we're actually trying to predict are the effects of changes in concentrations of individual enzymes and how that's going to affect the overall throughput. And so is that actually going to change the flux of things through the system? What we can see here is we might predict that metabolites and products would flow through these reactions differently based upon the concentrations of these different enzymes. Personally, I feel like one of the most interesting applications of this type of perspective is trying to figure out if we can understand the link of metabolism and biochemistry to the multi-omics data that we are collecting in all kinds of research projects and clinical contexts. Especially because if we can understand that link, we can have a much better understanding of what are the processes that are really going to help us understand, specific responses to perturbations of a system. And in other words, how do we tailor healthcare to reflect, the specific instance of a certain person in a specific case. And one of the really powerful tools in metabolomics that we could use is something referred to as a genome-scale metabolic model, or a GEM. And what this GEM is it is a collection of the biochemical reactions that occur in an organism, as many of them as we can. And this reconstruction is based on a wide range of high-throughput omics data and contains basically all of the known biochemical reactions that can be identified, and establishes a scaffold for the analysis of sort of omics level data. There are a lot of advantages to this genome scale metabolic reconstruction when you can do it. So in other words, reconstructing metabolomic reactions at a genome scale. You can get really good coverage of small monomeric molecules and do a really nice job of modeling sort of central metabolic pathways. They tend to be comprehensive network topologies, so they really are generating with all of the interactiveness and interactions between different modules and networks. But there are a lot of limitations to this approach. One of them is that if you're going to do this right, you probably need to do a fair amount of manual curation in order to create the pathways, especially those outside the central metabolism, which could be really important to what you're interested in. Additionally, there are some problems with modeling sort of polymeric metabolites that can be highly heterogeneous, so lipids, lipoproteins, things like that. But that said, I think that they're really dynamic and interesting tools, and I think speak to how we translate some of our genome scale data into usable predictive modeling outcomes. So if we think of like what this process actually looks like, is you have to take a whole lot of already established data and translate that into all of the gene-protein reaction associations and reactions that we would expect to see, right? So we have all of the protein reactions here. We've got our network, we've got the enzymes, we've got the proteins that go into them, and the genes that create them. right? So we have to generate this actual type of model. And in order to do that, right, typically to do this, we need to understand, what are the actual genes that are present, and what does it look like when they're expressed, and how are, these expressed genes then translating into enzymes, which are then affecting reactions. Once you have these associations, though, and you can define the relationships between genes and proteins and enzymes and reaction out and metabolic outputs, right, then we can create this kind of stoichiometric matrix, right? And in this matrix, basically what we have is every column is a reaction, and every row is a metabolite, right? So in this case, what we can see is that, if we have this column right here, right, these coefficients indicate the metabolisms that are going in, and the metabolisms that are being produced, and the metabolites that are being used, right? What's going in and what's coming out. And these are effectively the coefficients of the reactions in this process. And what you can see is that when you have a metabolic model like the one up above with all these different interconnecting pieces, the only way to really truly represent this appropriately is by generating this stoichiometric metrics of all the different reactions involved and all the metabolites and how they're interacting. Because as you can see, like some metabolites are interacting with multiple different reactions, right? Like they're involved in various different places. And so their abundancies and fluctuations are interdependent upon each other. And this goes back to those ideas about all those feedback loops that we talked about earlier. So if we think about, what are some of examples of these metabolomic, these Genomescale metabolic models, there's a lot of them out there now. They're generally increasing in number. We've seen an increase just, say, in like the human ones. We've seen Recon 1 come out in 2007, Recon 2 come out in 2010, Recon 2 come out in 2013, and Recon 3D come out in 2018. We can see that these gems are increasing in number and complexity as we go through time. And if we think about like some of the differences between these different versions of Recon, we can see, a steady increase in the number of reactions that are considered as part of this base model. Total number of genes has been increasing, but notice that total number of genes, because these are largely enzymatically based processes, right? It's way less than what we would consider to be, all the genes in an organism, right? But they are trying their best to not only include the number of reactions and the number of unique metabolites, but also looking at the role that three-dimensional structure and thus things like enzyme kinetics can have on the process of these data. And so there's a lot of information that can go into them. If you were to sort of conceptualize what it would look like, it would look something like this, these reconstructions, and this is what these networks would, like the complete network would look like. An example where we could see all this information start to come together is an examination of aryl sulfatase A, Arsa, which is used to hydrolyze cerebroside sulfate to cerebroside and sulfate. And mutations in this gene can lead to a condition known as metachromatic leukodystrophy, or MLD. MLD comes in a few different forms. There's a late infantile form, which is the most severe type of this disorder and the most common. Affects walking in children, leads to things like muscle wasting, weakness, muscle rigidity. progressive loss of blindness, convulsions, impaired swallowing, paralysis, dementia, and typically children suffering from this form died by the age of 5. There is also a juvenile form which has a somewhat later onset that has been shown to be related to things like impaired school performance and mental deterioration, dementia. And in this case, it's a slower progressing version of this disorder, but the age of death is just a little bit later in terms of 10 to 15 years from symptom onset. And then finally, there's an adult form of this disorder which occurs typically in like the 4th or 5th decade of life. Presents as a psychiatric disorder or progressive dementia, and generally is the slower progressor as compared to the infantile and juvenile forms. When we look at Arsa, aryl sulfatase, we can see the intersection between this sort of systems approach, this structural protein approach, and even understanding pharmacogenomic information. So if we look, you can find things like specific variants, sort of like this P426L variant, and is involved in some of the mechanistic progressions of the disease. So this type of variation, mutation, can lead to some significant changes related to the onset of ML, to be susceptible to MLD. What's interesting, though, is that if we look at where the majority of SNPs are, a large majority of the SNPs are actually located in close proximity to this metal binding site and substrate binding site for this protein around residue 306 and 309. And this shows that there's definitely a three-dimensional component where a lot of the mutations are going to be associated in this specific area, specifically the disease-causing mutations. And what we also tend to find is that, if we look at mutations that can occur, we can find that other proteins that intersect with sphingolipid metabolism also show evidence of having disease-linked mutations that are going to be overlapping or associated in these very similar spots. And so while Arsa is perhaps one of the most susceptible in terms of its effect on these types of genes, we can see that there's also some intersections with these other factors, indicating that what we're actually looking at is a network hotspot for these types of things, and that there can be a certain component where mutations in certain regions, which is based upon the structure and the functional role of some of these proteins, is particularly important. So we can start to see how the folding and the three-dimensional structure of proteins, mutations, their location inside of a network all contribute to our understanding of a system and the biological processes associated with it. But so that's kind of just looking at the structural components. What about thinking about when energy and molecules and activity are actually occurring within that network? And so, here we would take a series of methods that are typically referred to as Cobra Methods or constraint-based reconstruction analysis methods. And this is going to allow us to look at model dynamics. So before we were looking mostly just at topology, but now we're going to be looking at sort of the dynamics of the system. So what happens when we flux or push entities through the model? And so there's basically two different types of models that we would, or two different types of analysis we would do in this case: a flux balance analysis and a flux variability analysis. How this works is we need to understand, have a good understanding of how the underlying model is structured, right? So again, we would have a sort of theoretical network like this one, and these are slides taken from a presentation from Natal Van Riel about genome-scale metabolic models. And what they show, right, is these are the different players that we might see in these types of reactions. And so we have, if this is our hypothetical network, right, we've got our different reactions at each of these different cases. We have our different metabolites, 1, 2, 3, 4, and 5. And as I discussed before, this creates this kind of stoichiometric matrix, right, which is then can be derived into this stoichiometric model where we have each of the entities of this matrix basically representing the species concentrations or the reaction coefficients of these specific metabolites in each of these reactions. We then also can create a vector which would tell us what are the reaction rates, and assuming that we were looking at a sort of a steady state homeostasis situation, we can basically say that the metabolic balancing equation is going to be effectively equal to zero. And so based upon this process, we can actually derive and calculate, start to derive what these unknown rates of V might be, right? So we know they equal zero. We know sort of what the matrix looks like of all these equation coefficients. We can then go through and calculate what this V is here. One of the things that's important to note here is that there's not a single necessary solution for this. There could be multiple solutions to this type of question. And so what this ends up allowing us to do is to conduct a certain type of balancing analysis, right? What happens when this system is at a steady state? What's the dynamics that determine this system? And what we can do is we can start to identify, various different outcomes that could happen. So we can see whether or not we have, a specific, whether or not we have one specific outcome or two or three. We can start to generate what we think would be this particular space where we would find a metabolic fingerprint or indicator of what's going on in this system. And then we can try to solve this problem and figure out, what evidence we have to support these different types of modeling outcomes using something known as a flux balance analysis. So in a flux balance analysis, basically, we could have a diagram where we're trying to figure out, what's the best way, or what's the most representative or likely a way of resolving what's actually happening in a system. So we have basically two equivalent routes of producing metabolite, right? But we can sort of assume that, in actuality, that they're not truly equivalent, each of these two different pathways. Thus, we can say if we want to do something if our objective is to maximize the amount of ATP that is being yielded, and we know that there's only one route that can generate ATP, what we can do is we can link an objective function to this process that's either going to maximize or minimize the outcome that we want to have occur. So in this case, we're trying to maximize the output of ATP. So what we say is basically we create an objective function where the metabolic goal of each cell is for a specific condition, right? To create the most ATP, maximize biomass. This is a common thing, like have the most amount of growth, right? Like, so if we're going to have the most amount of growth, we need as much ATP. And how this process basically works is we start with no constraints, right? We've got this unconstrained space where we have all these possible solutions where any of the things that could happen, it could happen in any one of these locations. This is kind of this unconstrained space. And then we start to build in some constraints. So we could say, well, it's a steady state, right? So we're only going to allow it in this space, like in this positive space here, right? So we know that these V's are going to be positive, so they have to be through here. And then the next thing we can do is we can sort of create an allowable space for the solution, right? Where based upon these constraints, now we've kind of like limited the amount of this total space where things can be. And this is based on basically the stoichiometric matrix and some upper and lower bounds that we can pick that sort of become reasonable once we put those stoichiometric points and values into place. And then next what we can do is we can maximize for a specific function. So we can maximize, so if we know that this is the specific space in there, where in this space do we produce the most amount of, say, ATP, right? And we can find the specific point in there where that happens. So what this means is that we can end up with this really interesting situation where we can develop these genome scale metabolic models, either for, say, like a eukaryotic cell or for a bunch of different species of bacteria. And then we can do things like understand, like, okay, so we have this colonocyte. What happens if we start futzing with the different metabolites that are available to it? So let's take a case where, say, we have the metabolism of a normal cell versus a cancer cell. We know that, for instance, cancer cells are often highly dependent on glucose as an energy source. Can we minimize growth? Can we limit the growth of cancer cells, limit the replication of cancer cells, limit opportunity for there to be additional mutations by doing something like limiting the amount of glucose available to that cell? Can we use these types of processes to identify, where in this situation we might be? And we can do things like, and then compare it to a natural cell and see how the reactions might be different. Additionally, we could do things like use these types of analyses as well to understand the relationships in GEMs between different organisms and their interdependence. We can validate these processes by looking at reporter reactions or reporter metabolites to make sure what we're talking about is reasonable. And then we can even do things like take environmental samples to look at metagenome sequencing to calculate species abundances, and then build a physiological simulation model where we're not only considering the behavior of the cells in these situations, but also the behavior of the bacterial communities as well. So you can see how we can start to take all this stuff, and what's really powerful in this case is that we're able to scale up from a metabolic model all the way to start to make some predictions about how we think bacterial communities and cellular communities are actually going to respond. So if you think about it, a model is just a hypothesis, and this is one of the ways that we can use genome scale models to make direct predictions on what we think are possible outcomes in biological contexts of really complex systems. And we'll talk more about systems when we talk about systems biology, but it's quite a feat to be able to do this kind of thing. And by far, very complex. I showed you a lot of examples where there's a bunch of linear algebra and matrix calculations that you're probably not all that familiar with, but hopefully you were able to see and understand the general structure and context that is associated with these types of modeling approaches and conceptualizations of networks.